talked a lot about DNA so far, and we talked about the structure of RNA versus DNA, but we haven't actually talked about the secondary structure of RNA or even what RNA is used for. So in this section, we'll talk about RNA and transcription, which is how you make RNA. So just real quick, let's talk about the differences between RNA and DNA. So the first ones we've talked about before. Uracil is in RNA, thymine is in DNA, so those different bases. And then additionally, RNA has ribose, whereas DNA has deoxyribose. So those differences in the sugars. And then this is where we're starting to get a little bit different in the differences in terms of getting the new ones. But RNA is single-stranded. So we just talked about DNA is a double helix, meaning there's two strands of DNA. So RNA is only a single strand. And then additionally, RNA is much, much smaller. It's only in the thousands of nucleotides, whereas DNA is bigger, more like the millions. I like to think of this as an encyclopedia. So DNA is like the full encyclopedia or full volumes of the encyclopedia, whereas RNA is more like a single entry in that encyclopedia. But let's look at what types of RNA there are. So there's three major types. First one is ribosomal RNA. The second one is messenger RNA or mRNA. The last one is called transport RNA or tRNA. The first one, ribosomal RNA, is the most abundant. It's what most of the RNA in our body is. It combines with proteins to form the ribosome. But in terms of this course, we're not going to talk too much about what those actually do other than just knowing that they do something in the ribosome as ribosomal RNA. We're going to focus more on mRNA and tRNA. So mRNA carries genetic info from the DNA in your nucleus or in the cell's nucleus to the ribosome, which is in the cytoplasm. So DNA can only stay in the nucleus. But mRNA, messenger RNA, is like the postal worker who takes the data from the nucleus, copies it, and then takes it to wherever it needs to go. And then the ribosome is important to talk about because it's the protein synthesis machinery. So it's kind of like it's a machine that is going to kind of put things together. It's kind of like the proteins machinery to actually make proteins. Or enzyme machinery. So how you make mRNA is through transcription. I like to think of this as like prescription is relatable. A prescription is like where you write out what you're supposed to go and get. Transcription is where you're writing kind of a small instructions of codes of the DNA. There is a little bit of video on this. It's a very short video. I will post it alongside this video for you guys to see an animation of how this happens. But overall, what happens in transcription is that a portion, so this is the DNA here, these two strands, a section of the DNA unwinds using this RNA polymerase. RNA polymerase then attaches to one of these strands and is going to replicate that strand, but now using the nucleotides for our ribose nucleotides instead of the deoxyribose. It catalyzes phosphodiester bond formation. So very similar to how we replicated DNA, but now we have an RNA polymerase instead of a DNA polymerase. Replicate, take little kind of Legos building blocks, and then RNA polymerase is going to attach those all together. And the order is determined by those complementary base pairs. So the hydrogen bonding is going to help derive that order. And then at the termination site, RNA polymerase stops and mRNA is released. So once it gets to the point where it decides it's going to stop, RNA is released and your DNA just winds back together as it goes. Our last version of RNA is called tRNA or transfer RNA. It's the smallest in terms of actual size. It has the shortest number of nucleotides of all of the types of RNA. It adopts a T shape. So this is overall the shape of tRNA. And then it has a specific thing called an acceptor stem. This acceptor stem is on the 3' end, so it's this guy here. And what this acceptor stem does is it has a specific code at the beginning, and that code codes for bonding with a specific protein. So in this case, the AAC code means that it's going to be bonded with a serine residue or a serine amino acid. So an amino acid is attached at that acceptor stem. At the other side, there's something called an anticodon, which complements, which is a three nucleotide sequence that's a specific code. You'll see later when we're talking about genetic code that we're going to see what a codon is. So an anticodon is just going to be the opposite of that codon. So we'll talk more about that later too. But tRNA has an anticodon on it, which helps when we're talking about making proteins. We'll get to that in a minute. tRNA have different anticodons depending on the amino acid they code for. So this part of the anticodon is going to have, this particular anticodon is going to have a specific chain, or the overall primary structure is going to be specific to this anticodon. So if it's AAC here, it's going to be AGU here, kind of thing. And it's got a very specific chain. So this anticodon codes and says what kind of amino acid is going to be attached. So very specific amino acid is attached for the specific anticodon. And you'll see why that's important in the next section.